In August 2026, researchers at Stanford and the Arc Institute did something that had never been done before. They asked an AI to design a virus, not to study one, not to predict one, but to write its genetic code from nothing. Out of nearly 700,000 candidate genomes, 16 came alive in a lab. Real, functional, working viruses that had never existed anywhere in nature, invented entirely by artificial intelligence. This is how they built it, how it actually works, and why the people who study biological risk for a living are worried. Every virus that has ever existed came from evolution, mutation, selection, billions of years of trial and error. This one didn't. It came from a neural network trained on genetic code, the same way a language model is trained on text. Stanford bioengineer Brian High and graduate student Samuel King led the team that made it happen, and it worked well enough to kill bacteria that had already beaten a natural virus. To understand how this is possible, you have to understand what Evo2 actually is. It's not a chatbot. It's a genome language model, built by the Arc Institute and NVIDIA, with collaborators at Stanford, Berkeley, and UCSF. Where a normal AI model learns to predict the next word in a sentence, Evo2 learns to predict the next letter in a strand of DNA, A, T, C, or G. Same underlying idea, completely different alphabet. And because DNA only has 4 possible letters instead of tens of thousands of possible words, the model can pack an enormous amount of biological structure into every single prediction it makes. And the scale of what it learned from is hard to picture. Evo2 was trained on over 9 trillion individual DNA letters, pulled from more than 128,000 genomes spanning nearly every branch of the tree of life: bacteria, viruses, plants, animals, humans. It's 40 billion parameters deep, and it can hold a window of up to 1 million genetic letters in view at once, connecting patterns across distances no human researcher could track by hand. For comparison, that's roughly the same order of scale as the text datasets behind today's largest language models, just written in the 4-letter alphabet of life instead of English. This isn't a one-off moment either. Evo 1 shipped in early 2024. Evo 2 followed about a year later, with roughly a 10 times bigger training set and the ability to design far more complex genomes. This bacteriophage experiment used Evo 2 within months of its release. That's the actual cadence of this field right now. New capability, then a real-world lab result, inside a single calendar year, not a decade. Once a model understands the grammar of DNA that well, it can do more than just read it. It can write new sequences, 1 letter at a time, the exact same way a chatbot writes a new sentence 1 word at a time. Ask it to keep going, and it keeps generating letters that look and behave like real, functional genetic code, even though the exact sequence has never existed in any living organism, anywhere, ever. That's the entire trick. Reading became writing. So the team let it generate. Evo 1 and Evo 2 produced roughly 700,000 candidate virus genomes, far more than any human team could design by hand in a lifetime. Researchers narrowed that down to 302 worth actually synthesizing in a lab, using computational filters to guess which candidates looked most likely to actually function. Of those, 285 were successfully built into real DNA, and when they tested them against bacteria, 16 came alive as working functional viruses, a 5.6% hit rate, starting from nothing but an AI's imagination. Why did only 16 out of 285 actually come alive? Because writing a genome that looks statistically correct isn't the same as writing one that folds correctly, assembles correctly, and functions inside a living cell. A single wrong letter in the wrong spot can make an entire genome inert. Getting from a plausible-looking sequence to an actual working organism is still one of the hardest gaps in biology to close, which is part of why a 5.6% hit rate is being called a breakthrough, not a failure. These 16 weren't just any viruses. They were bacteriophages, viruses that hunt bacteria specifically and are completely harmless to human cells. A phage attaches to the surface of a bacterial cell, injects its genetic code inside, and hijacks the cell's own machinery to build hundreds of copies of itself. Then it bursts the cell open, killing it, and releases the new copies to hunt again. It's one of the oldest predator-prey relationships in biology, older than humans by billions of years. The AI didn't invent the concept. It invented 16 new predators, tuned to specific prey that had never existed anywhere before. This isn't just a lab curiosity. Antibiotic resistance already kills over 1 million people a year worldwide, and the pipeline of new antibiotics has been slowing down for decades. Bacteriophages are one of the oldest alternative weapons against resistant bacteria, used clinically in parts of Eastern Europe for almost a century. The bottleneck has always been speed. Finding the right natural phage for a specific resistant strain can take weeks. An AI that can design a phage on demand could turn that into hours, and they worked. Combined into a single cocktail, the 16 AI-designed phages wiped out 2 different strains of E. coli, strains that had already evolved resistance to a phage found in nature. In other words, the AI didn't just copy what evolution already built. It found something evolution hadn't, fast enough to beat bacteria that had already adapted. This part matters. The researchers deliberately trained the generative step on around 2 million bacteriophage genomes only. Viruses that infect bacteria, not people, animals, or plants. Sequences from viruses capable of infecting humans, animals, or plants were intentionally left out of what the model was allowed to generate, on purpose, as a safety boundary built into the project from day one. That was a deliberate choice, not an accident of the data. And it's exactly why the next part of this story matters so much. And Evo 2 isn't the only genome language model in development. Multiple academic labs and biotech companies are racing to build bigger, more capable versions of the same underlying idea, trained on ever larger slices of the genetic record. That race is exactly why biosecurity researchers are pushing so hard right now. While there's still only a handful of models at this scale, instead of dozens with nobody watching the door. Here's the problem. When you order synthetic DNA from a lab, there's supposed to be a screening step. Software that checks your sequence against a database of known dangerous pathogens before it gets printed. But that screening only works if the sequence resembles something already sitting in the database. A genuinely novel sequence, one that matches nothing anyone has ever seen before, can slip straight through undetected. And that is precisely the kind of sequence a model like Evo 2 is built to produce. Biology's version of a zero-day. Science ran this discovery alongside a companion editorial from Thomas Inglesby and Moritz Hanke of the Johns Hopkins Center for Health Security. Their conclusion was blunt. The governance to safely steer this capability does not exist yet. Today, most companies that synthesize DNA to order screen their customers only voluntarily, and plenty of orders face no mandatory check at all. Their ask is direct. Require every DNA synthesis provider, by law, to verify both the sequence being printed and the identity of whoever is ordering it, before a single strand gets made. So what actually happens from here? Inglesby and Hanke aren't just flagging a problem, they're pushing for specific fixes. Mandatory screening written into law, universal verification of who's ordering synthetic DNA, and screening tools that get updated as fast as models like Evo 2 do. None of that exists yet at a global, enforceable level. Arc Institute itself has said biosecurity review was part of the process before this was even published. But one research team choosing to be careful isn't the same thing as an industry-wide rule. That gap between one team's caution and everyone else's incentives is exactly where this story is headed next. If this feels familiar, it should. It's the same story that's played out everywhere else in AI over the last few years. Scale up the data, scale up the model, and capabilities appear that nobody explicitly programmed in. It happened with text, it happened with images, then it happened with protein structure, when AlphaFold solved a 50-year-old biology problem just by learning patterns in existing protein data. Evo 2 is the same idea pushed one level further, not just reading biology, but writing new biology that has never existed. And biology doesn't forget a capability once it exists. A few things worth knowing before the verdict. These are bacteriophages that target bacteria specifically, not viruses capable of infecting humans, animals, or plants. A 5.6% hit rate is still low. The overwhelming majority of AI-generated genomes did not become working viruses. This work was peer-reviewed and published in Science, with the biosecurity risks flagged in the very same issue, not buried in a follow-up nobody reads. The concern here isn't really this specific result, it's the general-purpose capability it proves is now possible. So where does that leave us? This is a real, peer-reviewed capability. AI can now design working, functional viral genomes from nothing but training data and a prompt. And the systems meant to keep synthetic biology safe were never built to catch something that's genuinely, truly new. Both of those things are true, and both of them matter. That's the story of the first fully AI-designed viruses. A real breakthrough, and a governance gap nobody's closed yet. All sources are linked in the description. Follow for more of these breakdowns.